Okay, so I think we're going to go ahead and get started. So to introduce our speaker today, I want to start by taking a look at some of her dreams. So you guys have all have dreams. One of the things you'll realize as you get older in life is that not all of your dreams are going to come true. So our speaker today wanted to be a cardiac surgeon. She also dreamt of being employed by L'Oreal Cosmetics, pulling down, I guess her dream was pulling down like a six-figure type salary. But those dreams didn't come true. She became a chemist. She ended up in a good place, even though her original dreams did not come true. Her work is what is considered to be provocative in her field. And I was wondering why she had the title of Proteomics Provocateur. Proteomics Provocateur. Does everybody here know what proteomics is? The study of proteins. So what could be provocative about that? Well, how many of you associate, what part of the body do you associate Alzheimer's with? The brain. So her work that she'll be talking about, dealing with aging and Alzheimer's, is going to actually question that, I guess, well-established idea that it is the brain alone. That's just a teaser. I'm not going to say much more. So it's a privilege and an honor to have Professor Robinson here with us today.
Thank you. Thank you, Dr. Murray, for the invitation. This is definitely, I'm very excited to be giving this seminar from the comforts of my own office. So hopefully, you all enjoy the things that we're doing in the lab as well, too. So to get us started, I thought what I'd do is just begin by highlighting some of the people that have been recognized nationally that have suffered from Alzheimer's disease. So there's former President Ronald Reagan, actress and just Hollywood TV and personality B. Smith, and then former coach, women's coach here, Pat Summitt. And if you know anything about Alzheimer's disease or other neurodegenerative diseases, then you know that they can be very devastating in order to watch people that are going through these diseases. And so the primary neurodegenerative disease that I'm going to talk about today is Alzheimer's disease. And it's a disease that we've seen manifested in individuals that have the disease. So they have issues with learning and with memory, with behavioral changes and things like that. Maybe you've had a grandparent that is has suffered or may be suffering from the disease, or know of someone that is. So it's very harmful and hurtful to watch. So what you may not realize is that there are a lot of people in the United States that are suffering from Alzheimer's disease. Currently, there are 5 million individuals that have AD. And it's the 6th leading cause of death amongst older individuals. And so it turns out that 1 in every 3 seniors dies of AD or some related dementia. So age ends up being the biggest risk factor for having Alzheimer's disease. And if you look at what's going to happen over the next 30 years, so down here you can see time, and this is the number of people affected with AD. It's predicted that the amount of people will go from 5 million to close to 15 million. So it's going to increase by a factor of 3 over the next 30 years. And, this is really important to get a handle on because of how much it costs to treat someone that has Alzheimer's disease. So there is no cure for the disease. There is nothing that can alter or slow the progression of disease. So the primary costs for taking care of someone that has the disease are just that. So it's $220 billion to take care of this 5 million set of people that have Alzheimer's disease. And that's something that personally I think about because I have older parents, and so thinking about if they were to suffer from this disease, you know, how that would affect my family dynamics and finances to be able to help care for them. So it is a brain-related disease, and we know a lot about some of the changes that take place in the brain. So if you look at the brain, a brain section from a healthy individual, and compare that to someone that has Alzheimer's disease, then you can see a noticeable difference in the size of the brain. So the individual with Alzheimer's disease has had a lot of shrinkage in their brain, and we call this atrophy. They've suffered from neuronal loss, so a lot of their neurons have died. And then they have these other changes that take place. So they have mitochondrial dysfunction, and then they have high levels of reactive oxygen and nitrogen species, which can end up being really harmful, and I'll talk about that later. The other two things that we know a lot about with Alzheimer's disease are the presence of these molecules that show up in the brain. So one of them is the presence of these amyloid beta peptides. And these peptides end up oligomerizing, so you have large aggregates of them. And in early stages of disease, or even in normal, healthy, older individuals, you'll see small depositions of these peptides that are present in the brain. But as the disease progresses, then the amount of these peptides and the location of them increases, and it spreads, so that if you look at the brain of someone post-mortem with AD, then you'd see a large presence of these senile plaques that are present in the brain. And this is also true for what are called neurofibrillary tangles, so tau tangles that show up in the brains of individuals with AD. Most of what I'll be talking about today looks at a mouse model of Alzheimer's disease that capitulates human AD by having the presence of these amyloid beta peptides present. So these peptides come about because you have this protein here, amyloid precursor protein, and it has a portion that sits outside of the cell membrane. And you can take these two secretase enzymes, beta and gamma, and if you cleave APP with these enzymes, then you generate these 42 to 40 amino acid long versions of amyloid beta peptide. So the mouse model that we use is one that also has these plaques that are present in the brain, and it's because we use human mutated forms of amyloid precursor protein and these secretase enzymes that we know lead to familial cases of Alzheimer's disease in people. So we know a lot about what happens in the brain. And one of the naive questions that I had as I was finishing my graduate studies and doing research as a postdoc had to do with the fact that, do we know that it's a disease that actually starts in the brain? Or could it be that something else outside of the brain leads to those changes? And it's an interesting question to think about, may not be as intuitive to think about. But what happens is that within the brain, you have immune cells or responsive cells in the brain, such as microglia and astrocytes, that end up helping to protect from different types of damage and inflammation in the brain. And these types of cells can send signals to the rest of the body. So if you're sending cytokines, for example, to different types of the body, then they would cross over this blood-brain barrier. So it basically is a gate that allows things in and out of the brain, and send them to other systems, either in the immune system or in other organs in the brain, or they just float through your blood circulatory system. The other thing that also happens is that you have other organs that can generate small molecules, large molecules, cytokines, or different types of cells that can then cross across this blood-brain barrier and send signals into the brain. And this happens because in Alzheimer's disease, this barrier here is broken and it's leaky. So things that shouldn't be able to permeate across the barrier end up permeating across the barrier. And what I found interesting as I was studying this was that tissues like the liver end up making those A beta peptides that end up being toxic in the brain. And so they're an additional source of amyloid beta. And there have been some groups that have shown that you can treat with different types of inhibitors that target the liver and seek to reduce amyloid beta peptide in the liver, but that they also end up reducing the levels of amyloid beta peptide that are in the brain. This has been demonstrated with a couple different types of treatment, and the results are somewhat controversial. However, it does show that there may be a role for other organs outside of the brain in the disease. The other thing that's interesting here is that the immune system of individuals that have AD or mouse models that have AD are impaired. And so if you look at different types of immune cells like T cells, they don't work as well, they're not as active, and this is demonstrated across the spectrum of mouse models to humans. So what I then had the goal of trying to pursue was trying to determine, one, whether or not we can use proteomics as a way to understand the relationship between changes that take place in the brain from someone that has Alzheimer's disease, as well as those that take place in the periphery. And then seeing, you know, is there a role for these other organs in the disease? And if so, what is or who are the initiators of disease? So can we figure out very early before disease onset occurs, what sorts of changes are giving us insight that someone is going to progress with Alzheimer's disease? So that's the thinking behind, you know, where our research program lies. So if you want to study disease, then you can do that in a number of ways. And so there are certainly groups that seek to look at the genetics, so looking at all the complement of the genes that exist in the organism, so that would be genomics. You can get information by looking at transcripts, so when you look at mRNA, or the study of all mRNA, then that would be looking at transcriptomics. But we become interested in looking at what we believe are the molecular players that actually do stuff in the cell. So genes are rather static, but proteins are very dynamic, and they cause action. And so our goal is to be able to study the entire complement of proteins that are present in a given tissue, cell, or a whole organism. And so this is what we mean by proteomics. It's like the study of all the proteins that we can detect in a system. And so there might be some students that are taking biology or learning about biology approaches to study proteins, but one of the major ones has to do with what we call a Western blot approach. And there the idea is that you can study one particular protein at a time. So here I'll represent one protein maybe by one pixel, or you can study several of these pixels which represent proteins that are involved in the same type of biological pathway. And what's necessary for those particular approaches to work is that you have to have an antibody that's specific to the protein that you're interested in order to study it and look at its levels in disease. So you have to have an antibody, which means that's a necessary thing. But in order to have an antibody means that you know ahead of time what protein you're interested in. So you have to have a priori knowledge about that. And this has been, you know, a very traditional state-of-the-art technology. However, it can be low throughput, meaning you can only look at, you know, one protein at a time. So we believe that in order to really get a full understanding of what happens in disease is that you have to be able to look at many proteins at a time. So if you want to get the complete picture behind what's happening, how do you go about doing that, and why is that necessary? And so we think it's necessary because the more information that you know, or the more that you can measure that's taking place in the system, then you can get insight about disease processes, how they're taking place. You can get information that might be helpful for discovering biomarkers, or you can get information about new targets that you can use for therapeutics. And so I like putting this picture up just because my hometown is actually in Louisville, Kentucky. And so what you may not be able to see without me giving you context is that if you look at these pixels very closely, then you can start to see an image appear. And so what you couldn't see before when you didn't have the entire picture in place, now you should be able to see one of the greatest boxers of all times. And he actually has 3 different expressions here depending on how you how you all are seated in the auditorium. So it's Muhammad Ali, in case you're guessing. Okay, so with proteomics, we seek to answer several questions. The simplest question has to do with what proteins are there, so how can we identify what proteins are there? But in the context of Alzheimer's disease, we not only want to know what's there, but how does it change between the two different conditions? Other questions that you can ask have to do with looking at what types of modifications have been added to the proteins. Is the protein function affected in disease? Where are the proteins localized with? What are the protein dynamics? So how does the protein fold, unfold? Who is it interacting with? So other proteins, is it interacting with small molecules? These sorts of things. So there are a host of questions that you can answer with proteomics. Really what we do in my group is we focus on these three. So what's there, and in order to address that, we use mass spec combined with liquid chromatography, sometimes electrophoresis. We care about how much something is there. So I'll share with you different approaches that we use for quantitation. And then we also have projects where we're interested in how specific proteins have been modified. So we look at oxidative modifications. The general way that we go after looking at proteins in disease is shown here. So what we do is we start with a tissue. This could be a postmortem brain tissue where we've gotten a slice of it from a human individual, or it could be looking at a liver tissue or heart tissue that comes from a mouse model of disease. And what we do is we extract all of the proteins out of that particular tissue. Once we've extracted all the proteins out of that, then we take the proteins and we use enzymes to cut them into smaller pieces. And so for each protein that we have in solution, we make about 20 to 30 peptides for that protein. So this peptide mixture ends up being really complex. So it has at least 1 million components in it or so. But we like making peptides because these are a lot more amenable to separate and study using mass spectrometry. So what we do first is we use multiple dimensions of liquid chromatography to separate out the peptides. So for example, we'll have one separation that we do where we separate based on peptide charge. We collect different fractions, and then for each fraction that we collect, we'll do a reverse phase separation that might be anywhere from 1 hour to 2 hours in terms of gradient time. So as components elute off of our column, what we're able to do then is send them into the mass spectrometer. And so the key pieces of information that we get from the mass spec are, one, this very accurate intact mass of each peptide. So we know this with high accuracy. And then we'll take each individual peptide peak and we give it a lot of energy and we break it apart in our instrument. And then we measure the masses of those fragments. And what's special about this particular spectrum is that we can look at the difference between mass of any two peaks, and it codes for one of the 20 amino acids. And so we can piece together what the amino acid sequence is for a given peptide peak. And then we use bioinformatics software to go in and help us identify how many proteins and what proteins we actually saw in our tissue with high confidence. So I won't go into a lot of detail, but I did want to show you a schematic of what the instrument looks like that we have in the lab. And so we have an LC system out here on the front end, and we use electrospray ionization to take our liquid sample and create a gas phase vapor of ions. And then we send these ions through a series of optics and electronics into what's called an Orbitrap mass analyzer. And we can get mass information out of this analyzer because the ions that come in, they end up orbiting around this, orbiting around this central electrode, and then they go back and forth. And based on how fast they're going back and forth between this region is relation to their mass to charge. So things that have low mass to charge move at really fast frequencies. Things that have higher mass to charge move at slower frequencies. So what we get back out is this first parent mass spectrum. And then this instrument actually has 2 mass spectrometers in it. And so what we do over in this portion of the instrument is we take individual populations of these peaks, and then we do those energetic collisions and make these MS-MS spectra. And you can go on and do an MS-3rd experiment. So the nice thing about it is while we're doing this measurement here and getting this data, at the same time, we're getting these fragment spectra over here, and we can get a lot of them. Okay, so we have ways that we can identify what proteins are there. And once we know what's there, we really want to know how much something changes. And so how do you do that? Well, one thing that you can do is not do anything to the sample. Just if you have 10 different samples, so 5 healthy, 5 disease, you can run them independently through a platform. And you can use the intensities of the peaks between your normal and disease and compare them to see how the relative concentration changed. And that's great because you can run an unlimited number of samples in order to do that. However, if you start to approach large numbers of samples, then it takes a long time to get through that because for each sample, you're independently running it. So a couple approaches that people use to get around that is to do either heavy isotope labeling of cell cultures, where you can add in heavy isotopes into the media of the cells as they're growing, mix them together. And in one single sample, now you have information for both your control and disease. Or if you're not working with cell cultures, then you can take the peptides that we make and you can add some specific type of chemical tag that incorporates heavy isotopes. And at that point, you can mix the 2 samples together, and then you can compare your control and your disease group. So these are nice because they allow you to increase your throughput by a factor of about 2 to 3. However, what happens if you want to look at multiple disease stages or you have many conditions that you want to study? And so one of the approaches that we take advantage of in my lab is one that combines, that uses isobaric tagging. And I'll show you an example of what that looks like here. So these are commercially available tags. One of the ones we use is a tandem mass tag, or TMT. And the structure of the reagent shown here. And what's important is that it has 3 different groups. So you have this N-hydroxysuccinimide ester, that's the reactive site. So this is going to react with our peptide amine groups. And then you have a mass balancer region and a reporter ion region. And in this reporter ion region and the mass balancer region, you can vary the heavy isotopes that are incorporated. So we can use nitrogen 15, carbon 13, or deuterium in this region represented by the asterisk, as well as in the mass balancer. So if you look at these 6 different versions of this tag, they all have the exact same mass, except if you look in this reporter ion region, here at 126, there are no heavy isotopes encoded. They're all here in the mass balancer region. And then the next tag at 127, we moved one of the heavy isotopes over here to nitrogen 15, and then reduced one of the heavy isotopes over here. So now, if you look at this portion of the tag, this would be 1 unit heavier than that one. So the way this works is you take 6 different samples, you react them independently with one of these 6 different versions of the tag, and then they bind to your peptides in your sample. You mix the 6 samples together, and you introduce one into the mass spectrometer, and what you see is 1 peak representing 6 samples. However, after you take that peak and break it apart, as I just described, then you break this bond here. And what that does is it releases this reporter ion, and it shows up at very low mass to charge values. And so we can use this as a way to indirectly measure the concentration of the peptide or protein in a given sample. And then we do that by looking at the peak heights. So the number of the sample tells us where it came from, and then the height of these peaks tells us how much it changed in a sample. So at the time that we began my lab, this was sort of where the state of the art was. You can mix 6 samples together. Now you can do this with 10 samples. But for us, the question really had to do with throughput and thinking about the total time to run these types of large-scale proteomics experiments. So even if I have the capability to mix 6 different samples together, but I want to study, let's say, multiple time points across disease and have enough statistics, so I'm going to take multiple animals in the same condition, then this still can become a very labor and cost intensive experiment. So we had the idea in mind that are there ways that we can increase our multiplexing capability? So can I get to a place where I can mix 50 or 60 different samples together, give them a unique chemical barcode, and then mix them together to run one or maybe just a few samples using the Proteomics mass spectrometry platform. So that's the goal and where we were headed. And so I'll, depending on time, I may be able to tell you about 2 stories of how we use that or just one. So this is our multiplexing technology and kind of how we use it. And so what we do is we take that mouse model that I described and we can study it in different stages. So if we look at young mice that are 3 months, they don't have any signs of disease. If we look at the disease model around 6 to 7 months, then it starts to have those amyloid beta deposits in the brain. It has some levels of oxidative stress that are higher than a wild type animal. And then if you look out here at 12 months or even later at 14 months, you have a lot of the senile plaques that are present in the brains of this animal. And then it very much behaves like a late stage Alzheimer's disease patient. So this is a mouse model that we created, and what we generated was a mouse tissue repository. So to study questions about the role of other organs in disease, what we have are brain, liver, lung, heart, kidney, and some other tissues that we've collected, and we have them at these different disease stages. And then we have our wild type animals that are pretty much normal, and then we have our Alzheimer's disease model. And for each one of these time points in genotypes and tissue, we have 6 independent versions. So if you look at all of this combined, what you see is that we have 180 samples that we need to analyze using our proteomics platform. And you know, here we think about the total time that it takes our graduate students to finish the program, and they think about that. And so to give a student a project, you know, with 180 samples can be very intensive. And so the student that was on this project said, well, we definitely have to get this multiplexing business to work. Otherwise, I'm never going to get out of here. And so what he came up with was this approach. And what it does is it combines the precursor isotopic labeling strategy I told you about, where you can mix 2 samples together, with the other strategy, isobaric tagging, that allows you to mix 6 together. So we can double the number of things that we can put together in a single analysis. So what we do is we take our wild type and disease animal. So we'll take the liver tissue, for example, from that animal. And then we have replicates of it. So these are 2 different sets of animals. And to this set of samples, we'll make the peptides. And what we do is we do a dimethylation reaction where we add a dimethyl group to the peptides, and we're able to target it specifically to the N-terminal amine of the peptide. And we do that because we modify the pH. And then what we do is we have another amine that's accessible on lysine residues of our peptides, and we tag those with one of those 6 reagents that I described, so that each one of these samples gets a different one of these 6 reagents. So that's 6. What we do over here to 3 separate pairs of wild type and disease tissues is the same thing. And the only difference here is that now when we do the dimethylation experiment, this one has carbon-13 and deuterium encoded in it. So we have 2 carbon-13, 6 deuterium. That tag is 8 units heavier than this version, but it still goes in the N-terminal amine. We save on reagent by taking the other half of our TMT tags, and we'll use them over here. And now we have all 12 samples uniquely tagged, mix them together, and then do our mass spectrometry. So I'll show you what this kind of looks like. So if you look in the first mass spec experiment, we should expect to see pairs of peaks that are separated by 8 Daltons coming from these heavy isotopes. We then take each one of these peaks, break it apart, get the sequence information to identify it. And then for getting really accurate quantitative information, we actually take one of these fragment peaks, and then we break it apart again, and we can get back out these 12 different barcode signals in the mass spec. Okay? So we've done this, and it works really, really well. So this is just an experiment where nothing should change across the 12 samples, and this shows you nothing changes. And then in experiments where we've intentionally modified the ratio or the amount of protein between 2 different samples in these dilutions here, then we can see that we can measure change accurately about to a 50 to 1 difference. Once the changes between peaks becomes more than 100-fold, then our ability to measure something accurately kind of goes down. Okay. So we've done this in multiple tissues, and this is data where we have done this in the liver tissue from the late stage Alzheimer's disease mouse. And so what's powerful about this particular experiment is that here we get information about one specific protein and how its level changes in our wild type and our disease animal, but we can compare that across 6 biological replicates from each condition in a single experiment. So we've saved a lot of time and a lot of expenses by being able to do this compared to if we had to run 12 separately. And what we do is we look at what's actually really different between the samples. And so in this case, we identified 1,700 proteins to start with. We were able to get good quantitative information from a little more than 1,100. And so what we do is we compare the intensity of the peaks from our disease animal to our wild type, and we plot that as a log-2 ratio value. And so this tells us how much it changes. And then what you see here in this plot are, these 1,100 different proteins, but the ones that have the most statistical differences between our wild type and disease animals are shown here in red. And so that ends up being about 64 proteins. Okay? The other thing that I'll just highlight briefly is that if you look at this axis, this tells us the total amount of signal that we measure across these channels for each protein. And this is important because if you think about working with a really complex sample like liver tissue that comes from a mouse or brain tissue that comes from a human, the ranges of concentrations of proteins in that sample is tremendous. And so it's over 6 to 12 orders of magnitude. And so with our technique, we can see proteins that differ a lot in the concentration, and then we can also look at not only individual variation, but also biological variation across disease genotypes. All right, so just quickly, we verify that our approach is valid also by doing a duplex experiment. So here what we do is we do our demethylation in such a way that now we target both the N-terminus and the lysinamine, and we'll take pairs. So we'll take a wild type and an AD sample, do the tagging, mix them together, and now here we have 6 samples. Okay? So we compare the duplex experiment to our 12-plex, and what you see here are individual proteins where we can look at their changes in our disease animal compared to the wild type. So however these proteins changed in our 12-plex experiment, they change in a similar direction in our duplex experiment. So by 2 different quantitative platforms, we are very confident of the changes that we can measure in our Alzheimer's disease animals. So one of the things that we do to evaluate how these changes tell us something about the disease process is we look for different types of biological pathways. And so one of the things that happens in individuals that have Alzheimer's disease is shown here. So if you look at a PET scan of a healthy individual that's age matched to someone that has Alzheimer's disease, is that the levels of glucose are a lot lower in the individual that has Alzheimer's disease. So we call this hypometabolism. And so as the metabolism or glucose metabolism in people with Alzheimer's disease goes down with time and disease progression, their cognitive impairment goes up and their neurological function also goes down. Okay? So these are well-known phenomena that happen in the brain. However, one of the things that we see in our data is if you take that 64 set of proteins and you look to see, what pathways they represent, we saw a lot of proteins that are involved in fatty acid metabolism and beta oxidation that are higher in expression in our disease animals. And they end up leading to acetyl-CoA, which ends up leading to the formation of these ketone bodies. And these ketone bodies are these small molecules that are shown here. So beta hydroxybutyrate, acetoacetate. And these are small molecules that show up in your body whenever you're under conditions of starvation. So you don't have like a lot of glucose around. They show up as an alternative fuel source. So we see evidence for what we believe is the production of high levels of these ketone bodies in the liver of an AD mouse model. And we think that they're being transported across that leaky blood-brain barrier to the brain to compensate for the fact that in the brain, the glucose metabolism is being altered. So this is one of many stories about different biological pathways that we collect in my lab with different types of data. Okay, and so I have a whole team of undergraduates and graduate students that are studying lipid metabolism and AD. All right, the second story that I want to share with you before you head out is one that has to do with modifying our approach to be able to study different types of modifications to the proteins in disease. So these are what we call Redox proteomics experiments. So one of the things that we look at is cysteine. So cysteine is an amino acid that has this thiol group out here. And cysteine can undergo a number of reversible and irreversible modifications. So if you have these reactive oxygen or nitrogen species that interact with cysteine, you can form these irreversible acidic forms of cysteine, or you can form any number of these different types of reversible modifications. So a common modification of cysteine is the disulfide bridge oxidation state, so extra between two proteins or within a protein. Or you can have modifications like S-nitrosylation or S-glutathiolation. So there are a number of different types of these modifications. So they're very dynamic, they're very labile, they're versatile, there's a good number of them. And some of these modifications are very helpful for the cell being able to just do normal signaling in business. However, when you have really high levels of these reactive oxygen species, then this can be very detrimental to cysteine. And so because cysteine is oxidation prone, then it shows up as being modified a lot in disease. And so we're interested in the role of these cysteine modifications in aging and especially in Alzheimer's disease and how they affect protein function. So I won't go through all the challenges that we face looking for this modification, but I'll just give you one major one if you look here at this color grid. And so if we take our peptide sample where we have all of our peptides and we only consider the number of peptides that contain cysteine, it's 14% of the entire proteome. And then if we consider the number that can be reversibly oxidized, then the levels are about 1% of this 14%. And then if we consider just 10 different types of oxidative modifications to cysteine, then that would mean that each one of these modifications exists at a level that's about 0.1%. So what we're really looking for in trying to study these modifications is like a needle in the haystack because they're very low in concentration. Nonetheless, we think they can be very important for helping us to understand signaling in Alzheimer's disease. And so one of the modifications that I'm going to share that we work with is S-nitrosylation. So S-nitrosylation is a post-translational modification of cysteine that under normal conditions in the brain is very critical. So it's critical for neuronal plasticity, differentiation, neuronal development, and for neurons to survive. However, one of the other things that can happen with this modification under conditions of oxidative stress is that it leads to protein misfolding, mitochondrial dysfunction, severe endoplasmic reticulum stress, and then your neurons become damaged. So on the one hand, in Alzheimer's disease, it can be very neuroprotective, but on the other hand, it can be very neurodestructive. And if you want to start to answer questions about in which situations is it helpful versus harmful, you have to have methods that allow you to identify all of the proteins that actually have these modifications and localize where the modifications are. So we take our C-pilot approach that we started with earlier that allows us to multiplex 12 different samples. And what we've done, in short, is to modify it to only focus on nitrosylated proteins. And so what we're able to do is we're able to take our wild type and Alzheimer's disease samples, and we can look for these oxidized cysteine residues in the sample, and we can go after them. So what we do is we can block these cysteines with some blocker, the ones that are free and not modified first. So we use this reagent N-ethylmaleimide. And then what we do is for the ones that have S-nitrosylation, we reduce them with ascorbate. And so what ascorbate does is it takes this SNO group and turns it into an SH thiol group. And so once we do that, that allows us to use these resins, and these resins can capture these free thiol groups. And so we capture the peptides that have the cysteine thiols free on the resin, and then when they're on the resin, we do the N-terminal and lysine tagging as I described before. So what you have here in a single experiment, although this may look complicated where you sit, is we have our wild type and disease samples where we can get information about just the total cysteine levels in those proteins. And so what we do here is we reduce everything and just look at total cysteine. And then what we do specifically is take our wild type and disease animals. So here we have one wild type, one disease, one wild type, one disease. And then we look for the S-nitrosylation by doing that selective reduction. So in our single 12-plex experiment, we're monitoring 4 different wild type and 4 different diseased animals looking for this S-nitrosylation level. All right, so this just shows you how we look at the data. So we get the MS information, do the MS-MS experiment, and then what you see out here are these reporters that tell us about 126 and 127 are our total cysteine levels, and then out here, if this cysteine was modified, we would see peaks show up between 128 to 131. But if you look here at this cysteine 96, in the literature, we looked it up to see if there's anything known about it being modified and modified in Alzheimer's disease, and it's not. So this cysteine belongs to peroxyredoxin, and it shows up as an SOH modification. So we wouldn't expect or shouldn't expect to see any signal showing out here in that region. However, for other proteins like this one, so this ROS-related botulinum toxin substrate protein, then we can start to see some signal out here. So this is telling us that this modification on this very specific cysteine in the protein shows up in our wild type animal and our disease animal, or wild, not in this wild type, but in this disease, and then so on over here. So, you know, we can get information about where the modification is, what level it shows up in wild type and disease, and we take advantage of having this total cysteine information to help us normalize the level of how the oxidation is changing. All right, so I know I need to wrap up, but what I will tell you is that, you know, what we've been able to do with this characterization of these modification sites in disease has been pretty impressive. So in the literature, looking at mouse and human models, there are these handful of reports that have identified anywhere from about 38 to 138 different SNO modified proteins. So the total number of these modifications is, right now, our ability to see them is a lot lower than looking at other things like phosphorylation. But what we've seen are 56 consistent sites with these other studies, and then we've identified 79 new S-nitrosylation sites that that have not been reported before in Alzheimer's disease. And so here we're looking at Alzheimer's disease brain tissue, and what we find are 93 SNO modifications that show up in both wild type and our disease animals. And so we've been going through looking at what these modifications are involved in. A lot of the proteins end up being involved in signaling processes that are necessary in the brain or in metabolism processes. There are also things involved in the immune system and other types of changes that I won't get into. But this is very promising for us because, one, we've been able to detect some very important things in disease. And so some of these proteins change by very little between the wild type and the disease animals. Others change by a huge factor. And so one of the ones that we've become interested in are these 14-3 gamma proteins, or isoforms of gamma proteins, because they have really high levels in terms of how they are modified. So not only are they modified, but they're modified in high levels, and they have a substantial change. So it's a factor of 2 change difference in level between our disease animals and our wild type animals. So we have looked at this modification in disease, and I'll just start to close by sharing like some of the other ways that we're going after addressing the problem of how to study the role of different organs in Alzheimer's disease. So here's the work of Christina, and what she's doing is she's using our multiplexing approach to study in a single experiment the changes in protein expression from heart, brain, and liver and across wild type and disease animals. And this is pretty cool because what you can do is you can look at, for instance, all the proteins involved in the glycolysis pathway, and you can start to see how they change in an individual tissue between wild type and disease and follow that across different organs. So then you can see whether or not there's any relationship between protein changes across tissues and investigate what the relationship is between brain and these organs outside of the brain. So we're excited about what she's doing in the lab. And in order to make all of these experiments work requires having a lot of informatics in place. And so our data sizes that we generate are fairly large. So on average, anywhere from 60 to 70 gigabytes. Here, in her experiment, she identified 4,800 or so proteins. And so we have undergraduate students that even help us with our bioinformatics analyses. So we're coming up with a bioinformatics platform to really streamline getting through all of our data analysis. We have other projects in the lab where we're working to reduce the cost of all of these experiments. So the tags that I told you before, these commercially available tags, couple $1,000, so about $500 per experiment. So we're working with a collaborator, Ling-Jung Lee and Dustin Frost at Wisconsin-Madison, who synthesize their own tags in-house. And so this is their version of the isobaric tag Dilu. And really the important thing about their tag is that if you look underneath any one of these reporter ions and you have a really high resolution mass spec instrument, is that what looks like one peak is actually 4 different peaks. And so if you do that for each one of these peaks, we can take our multiplexing from 12 samples to 24 samples in a single analysis. And so this is just some preliminary data where we've done that with yeast. We also are working on developing our own tags, and so we have summer undergraduate students who have come into the lab and helped us there. We're continuing to work on the Redox work. We'd like to automate our entire process. And then we have a whole team of students that are working on using different types of chemistries to further enhance our multiplexing abilities. So let me just close by sharing that I hope that you realize that, one, the technology development that takes place in my lab is very important for increasing our multiplexing capabilities. I hope that you've been able to appreciate how we can use these abilities to study the proteome in Alzheimer's disease and begin to answer some questions that we have about differences between brain and other organs. And already we've been able to show using our global platforms and with Redox proteomics analysis that energy and lipid metabolism, as well as oxidative stress, seem to be really important pathways that exist not only in the brains of individuals or our mouse models that have AD, but also in peripheral tissues. And so I'd like to thank all of the students that contribute to our work. This is a list of our current members. These are our group alumni, including a large cast of undergrads. And then we have a lot of important collaborators, especially at Wisconsin, Madison, Minnesota, that are helping us with software. And then our collaborators at the Alzheimer's Disease Research Center here at Pitt that help us with sample. And then I'd like to thank NIH, who's currently funding this work. And I'm more than happy to entertain and answer student questions. So thank you all again.
Okay, remember to say your name.
Hi, my name is Kadra. You mentioned that there was a group of students working with metabolism, specifically dealing with ketone bodies. Do you know if they've discovered anything beneficial for Alzheimer's other than what you've mentioned already?
Yeah, so for this group of students, so one, let me just say there's a paper, or there are a couple of papers that have come out from other groups in South America that have actually looked at metabolites in these same tissues in Alzheimer's disease. And so they've been able to show that these ketone bodies are higher in these other tissues in Alzheimer's disease compared to healthy. and so that's been an important finding because it like corroborates our story, but looking at the protein level. The students in our lab that are working on lipid metabolism, so far I wouldn't say that they've fully identified things that's helpful to disease because they're still working on method development and running experiments. And so one of the ways that we're studying lipid metabolism is by developing platforms to look at lipids in Alzheimer's disease. So we have students working on doing that in plasma tissue that come from people. And then we also are studying lipid metabolism in the context of other diseases. So individuals that have Alzheimer's disease are at higher risk of having other things like heart disease or obesity or diabetes as well. And all of those diseases share a common thread of lipid metabolism being off or dyshomeostasis existing in lipid metabolism. And so my group is studying some human plasma samples from individuals that have these other conditions as well as Alzheimer's disease. So right now it's too early to say, but I think give us a year or two and we'll be able to say a lot more. Thank you for your question.
Hi, my name is Helen, and I just had a kind of a general question about how you envision your work with protein analysis tying into the treatment of Alzheimer's, and if you were partnered with anybody who is working on that side of things and taking your data to synthesize different ways for treatment.
Yeah, so right now we're not at the point where we have like a specific partner. We are part of our Alzheimer's Disease Research Center, where they have like ongoing different types of clinical trials with different drug therapies that they're beginning to come out with that they're testing. So for us, what we're trying to do is understand like how the disease is manifesting. And so in the proteins that we see that are changing and that change consistently in our mouse system and in human tissues that we've investigated, we're able to take a smaller list of proteins that we think are important for disease and start to share that with the Alzheimer's disease community so that if people are looking at developing drugs for new types of targets, then hopefully the information that we're generating is helpful in that regard. So yeah. Good question.
Hello, my name is Dylan Zimmerman. You mentioned earlier how you're using a multidimensional liquid chromatography separation method. My general question, have you maintained that methodology for all the proteins, or have you sometimes gone to even more dimensions like 3 dimensions or 4 dimensions?
Yeah, so it can get pretty crazy the more dimensions you add. So we generally do like three dimensions, so a couple of dimensions of chromatography, and then the mass spectrometry, we have at least two dimensions that we do there. We have done an additional separation sometimes in the mass spectrometer instrument where you can separate out peptides in the gas phase. So we call that gas phase fractionation. And it's a way that we can run the instrument to separate out in the mass dimension first. And so we'll combine that with the multidimensional LC platform and then the gas phase MS platform and then the MSMS and the MS to the third. So our experiments end up being very multidimensional. It really just comes down to us having to trade off like how in depth do we want to investigate a particular proteome sample versus how much time, reagent, and resources we have in order to do the experiments. So sometimes we don't do any multidimensional work. We just do like one LC separation and then mass spec because we need a very quick answer, but we're only probing like the surface level proteins that are changing. Good question.
Hi, my name is Yewan, and I just had a question generally about Alzheimer's. And you said that people with Alzheimer's generally have a higher chance of having other diseases such as liver disease. What about Alzheimer's is it that ultimately leads to it being a high cause of death?
So with Alzheimer's disease, it really sets people up for different types of challenges. So like one of them might be issues with having compromised immunity. So sometimes people that have Alzheimer's disease can die because their immune system's compromised and they get, some sort of pneumonia type infection or something like that ends up maybe being the final cause of death. But I think anytime that your neurons are continuing to die and your brain is being less active, that has effects overall on your entire, body, being the final cause of death. It also is what do you call it? Inverse relationship, I guess, as well. So individuals that have heart disease and type 2 diabetes and cardiovascular diseases, like these individuals also are at a higher risk of developing Alzheimer's disease. I don't think we know like fully why those relationships exist, but definitely, lipid metabolism and lipids seems to be a pretty important pathway that is altered in both sets of these different conditions. Did I answer your question?
Yes.
Yes, okay, thank you.
Hi, my name is Jason Grellman, and I know Alzheimer's can often affect long-term and short-term memory differently. I was wondering if the proteins or mechanisms that you've been looking at kind of explain how that works or anything.
Yes, I wouldn't say directly the things that we're doing in my lab now, but some of the things that I studied as a postdoc where we were looking at more human brain tissues, as well as other groups in the field that are looking at in brain, definitely being able to look at all these different biological pathways and the proteome allows us to make some connections with how proteins are changing, for example, in specific types of signaling pathways with the correlation of neurological function or cognitive impairment. So there are a number of groups that start to make like those connections. So yeah, there's a whole, whole array of literature that exists on that specific topic. And there's still a lot to be learned because we don't have it all figured out. Yeah, it's really complex.
Hi, I'm Gerald Bahimba. This is kind of more of a personal question. It was mentioned before that you wanted to go into cosmetics and surgery. So what is it that really made you change your mind to do something different?
That's a good question. So, so I had a change of heart as a undergrad student about the whole med school thing. I went into grad school wanting to do cosmetics, but I got roped into working on this aging project and learning about technology and how you can use chemistry to study disease opened up a world of new possibilities for me. I really liked some of the freedoms that I had as a grad student to explore research in detail and just learn new things all the time, as well as a postdoc. So when I interviewed for like industrial jobs and things like that, and when I did, I don't know, I just didn't feel like I would be very satisfied long term in some of the positions because I wanted to have the freedom to explore. And so, yeah, between that, I think there was a lot of connections of my research as a graduate student, like the things I found. So I worked in flies as a grad student in aging, but I saw connections with what I was learning when I was working with human tissues that came from people with Alzheimer's disease. So being able to see those common threads, I really saw that there was something important to keep studying. And then I'd just say the final thing was that I had a really great graduate advisor and also a postdoctoral mentor who really showed me just the importance and value of how you can use chemistry and make an impact in things that are important like disease. So for me, I'm very driven by the fact that people are suffering from the disease, and I could be one of those people, one day in the near future, and my parents could be one, like really soon. That motivates me and drives me. So being able to use all these skills and knowledge that I've invested basically my whole life being in school, just kind of, I found my purpose, I guess, is the short of it. Does that help answer your question?
Yes, it does.
Okay, good. And I'm happy to talk more offline too.
My name's Michael. Could you just briefly summarize what's happening on the mitochondrial level with the brain during Alzheimer's?
Yeah, so there's a lot of mitochondrial dysfunction. So there's issues with electron chain transport that are altered in Alzheimer's disease, and those changes become more pronounced with disease stage. That becomes really important because it affects energy metabolism and ATP synthesis and all these sorts of things. And then it also lends to the generation of these reactive oxygen and nitrogen species that end up being present in really high concentrations in the cells as well. And so because there's this mitochondrial dysfunction, then that leads to all of these proteins, DNA and other molecules being oxidized and damaged. And so it really just is like a domino effect, where once you have all these ROS that are being generated from the leaky mitochondria, then all of that perpetuates in damaged proteins and pathways that end up being blocked or not functional, which then manifest in the behavioral things that you see in people, as well as neurological dysfunction.
Okay, we want to thank you very much again. Yes.
Thank you all. Thank you all for sticking around.
So are you going to be looking for high school and undergrad students for the summer?
Certainly, so students can feel free to check out our website, razorlaboratory.com, or email me directly if they're interested in opportunities.
Okay.
We have a couple of summer programs in our department that students may be able to take advantage of, and I'm always interested. Yeah.
Okay, I'll send them your information.
Okay, thank you,
Okay,
Desmond. Thank you for the invitation.
All right, thanks.
Okay, bye.